Good morning. I would like to thank first the organizers for allowing me to present our work here. And I'm going to discuss with you today our work on decoding the STAT5 activation and repression code for biomarkers discovery. I'm going first to talk to you a little bit about the biology of STATs and STAT5, and then I'm going to present our work on identification of novel interacting partners of STAT5, and I'm going to show you some proteomics, some genomics, and some transcriptomics data. Then I'm going to discuss how the interactions influence STAT5 dependent transcription and how STAT5 transcriptional networks can be used for biomarkers discovery. The signal transducers and activators of transcription, STATs, are encoded by 7 STAT genes in mammalian organisms: STAT1, 2, 3, 4, 5A, 5B, and 6. STATs participate in normal cellular events such as differentiation, proliferation, cell survival, apoptosis, and angiogenesis. STATs contribute to signal transduction by various cytokines, hormones, and growth factors, and STATs are activated by tyrosine phosphorylation. Inappropriate STAT activation has been described in many solid and hematologic malignancies. STAT5 is encoded. The specific factor with which we work is encoded by 2 genes, STAT5A and STAT5B, which share 96% homology. STAT5 regulates genes involved in response to cytokines and growth factors like the cell cycle progression related genes in hematopoietic cells or genes related to mammary gland milk production. Constitutive STAT5 activation in mice causes leukemogenesis and tumor formation, and in humans causes solid and hematopoietic malignancies. Upon binding of a ligand to its receptor, STATs are phosphorylated, dimerize, and translocate from the cytoplasm to the nucleus, where they bind to specific regulatory elements and tightly control gene transcription. The objectives of our research is to understand how STAT5 is negatively and positively regulated, and how protein interactions and target genes expression change at specific time points. So to this specific project, we try to understand which are the interacting partners of STAT5 in the nucleus of probe cells, and how the interactions influence STAT5 dependent transcription and which target genes are expressed. Our future goal is to define biomarkers and therapeutic targets for hematologic and solid malignancies. We used probe cells, which are IL-3 dependent, to identify proteins interacting with STAT5 Genomic targets of STAT5 and of the interacting partners with chip sequencing, and changes in STAT5 target genes expression with RNA sequencing in wild type and in cells with knockdown of STAT5 or of the interacting partners. At the end, by various bioinformatics approaches, we analyzed the data, we integrated the data, and we tried to understand how STAT5 transcriptional networks are controlled. So we applied a method of in vivo biotinylation in our cells. We tagged STAT5 with a biotag and we co-expressed this construct with a BRFAA biotin ligase, which inside the cells biotinylates biotag. Thus we have a biotinylated protein which is easy to be isolated with high affinity streptavidin bits. In this way, we isolated STAT5 protein complexes and performed mass spectrometry to identify potential STAT5 interacting partners. In total, we obtained 711 proteins, and from those proteins we selected LSD1, a histone demethylase capable of either gene repression or activation, and histone deacetylase 3, which HDAC3, which is a histone deacetylase leading to gene repression. The first step of our work was to validate the interactions of STAT5A with LSD1 and HDAC3 with immunoprecipitation, streptavidin bits pull down, size fractionations, and various other biochemical experiments. For STAT5A, in IL-3 stimulated cells, we obtained around 15,000 STAT5A peaks, genomic regions that bind STAT5A, which were located in introns, in intergenic regions, and in a smaller percentage in promoters. We then took all these targets and we performed RNA sequencing experiments in wild-type cells and in cells with knockdown of STAT5A, STAT5B, LSD, and HDAC3. And these experiments showed us that strong STAT5A binding correlates to gene activation. You can see here that the red is upregulation, the green is downregulation, and strong also STAT5A binding correlates to gene repression in STAT5A and STAT5B knockdown. And also most STAT5A targets were upregulated in the absence of LSD1. Here you can see red. We then plotted STAT5A binding sites upon IL-3 stimulation, and again we checked if there is colocalization for LSD1 and HDAC3 at these sites. We plotted LSD sites upon IL-3 stimulation and upon IL-3 starvation, and also HDAC3 sites on IL-3 starved and stimulated cells, and we checked all the colocalizing sites all over genome. and we also plotted the RNA sequencing data and checked the expression levels of the genes on these close to these specific genomic sites. We then performed clustering of the chip sequencing, the genomics and the transcriptomics data, and defined groups of genes with different binding of STAT5A, LSD, and HDAC3 in terms of strength. So we defined groups with strong STAT5A binding and weak LSD1 and HDAC3 binding, less strong STAT5A and strong LSD and HDAC3 binding, less strong STAT5A and weak LSD1 and HDAC3 binding, and only STAT5A binding. And then we performed motif analysis, and in all cases we detected the canonical TTC GAA STAT5A motif, and only in 2 clusters with strong LSD and HDAC3 binding and weak LSD and HDAC3 binding, we detected the half GAA STAT5A motif, and these clusters were in a higher percentage located in promoter regions. As an example of how all these target genes can be used in translational research, I'm going to show you some data of our work on a specific STAT5 target gene we identified in the past, which is called GPF3, and which is an epigenetic key factor associated with a BAF chromatin remodeling complex. And this gene, when we tested its expression levels in various patients with hematologic malignancies, had an upregulated expression in BCLL, B-cell chronic lymphocytic leukemia. And we are currently testing the expression levels of this gene in grade 1, 2 and grade 3 breast cancer samples. And so far we see an upregulation of this gene in grade 3 breast cancer. Our conclusions are that we identified novel STAT5 interacting partners, LSD1 and HDAC3. We identified common genomic target genes of STAT5A, LSD1 and HDAC3, and there is a dual role, activating or repressing role of LSD1 and HDAC3 in STAT5 dependent transcription, and the direction of transcription is determined by protein interactions, binding affinities, motifs, and genomic positions. Our current work and future plans, of course, involves testing of expression levels of the target genes identified and of the changes in the networks of target genes in adult and pediatric patients with hematologic malignancies. And such genes can be used as biomarkers for prognosis, diagnosis and stratification, for selection of optimal treatments, and for follow-up after the treatments. Furthermore, the targeting of STAT5A interacting partners will be important for development of novel molecular therapies. I would like to acknowledge the people that were involved in this work: Katerina Nanu, Χρυσαυγή Τουμπέκη, Vasiliki Lazu and Marina Theodoru, and our collaborators from our institute, Dimitris Thanos, Matt Lavigne, Triadaphilos Paparoudas, from Erasmus Medical Center Jeroen Demmers, and from University of Thessaly Mattheos Peletas and Antigoni Mamara, and from the University of Athens Georgia Pachristopoulou and Andreas Korillas. Most of the work I presented is published in NAR and in PLOS ONE, and I would like also to thank EU for the funding, Research Promotion Foundation, and GSRD. Thank you for your attention.